Okay. So there might be cases where we want to know probabilities when we're dealing with even more than 2 traits. Okay. And so in this case, we probably definitely would not want to have to make a Punnett square for 3 traits. That'd be very large. So we're going to practice using, again, those rules of statistics, of probabilities to answer these questions. So we'll do the first one together, and then I'll give you some time to talk about the second 2 with your group. But I really do encourage you to write this down in your notes, because there will definitely be a question on the test that is like this. So, depending on how good your skills are with monohybrid crosses, you may or may not want to make the Punnett squares out. I'm going to do it just since we're doing this all together. So, get a marker that works. So we'll do a Punnett square for each one of those crosses individually. So for gene A, parent 1 is heterozygous, parent 2 is homozygous recessive, right? So our genotype ratio is going to be 1/2 heterozygous, 1/2 homozygous recessive, right? We take a look at the second trait, the B trait. Parent 1 is homozygous dominant. Parent 2 is heterozygous. Our genotypic ratio there is going to be 1/2 and 1/2, right? 1/2 homozygous dominant, 1/2 heterozygous. And for our last trait, C, parent 1 is heterozygous, and parent 2 is homozygous dominant, right? So it's also going to be 1/2 and 1/2. 1/2 homozygous dominant and 1/2 heterozygous. So you already have all those. If you can look at any monohybrid cross and already know the genotypic ratio, then that makes this a lot easier. If not, you can always set up the Punnett square. So letter A, we want to know the probability of the offspring being heterozygous for every single trait. So if we know our ratios for each individually, how are we going to determine this? What's the chance of them being heterozygous for A? 1/2. Times, what's the chance they're going to be heterozygous for B? 1/2. Times, what's the chance they're going to be heterozygous for C? 1/2. For a total of so go ahead and talk through the next 2 with your group real quick. Okay. So letter B, what's the chances of them being homozygous recessive for A? And homozygous dominant for B? And heterozygous for C? So that's ends up being the same, right? 1/8. For letter C, instead of thinking about the genotype ratios, you have to look at the phenotype ratios. So what's the probability that they're going to show the dominant trait for A? 1/2. What's the chance that they're going to show the dominant trait for B? 1, all of them. And C? 1. So 1/2. Okay. Half of the offspring from this cross are going to have all the dominant traits. Okay, so if it asks you a question like this, and you don't have to draw a huge Punnett square, you don't have to draw a 4th-line diagram for everything. You can just calculate them individually like this. Okay, so any questions on that before we move on? Like I said, there'll probably definitely be a question like this on the test. So just make sure you're able to apply those probability rules, right? Just multiply the probability in each case together, and that's going to give you the total probability. Okay, so we're going to be moving away from simple Mendelian genetics today to some other types of inheritance. So if you remember anything about like incomplete dominance or codominance in bio 1, that's what we're going to start looking at today. So again, I would just kind of make sure you're taking notes as we go through this. We'll do some practice problems along the way. Maybe I'll just hand this out now so I don't forget. I don't know how many of you, I know quite a few of you are juniors, so you're probably going to have to be here tomorrow for ISTEP testing. What I'm going to pass out is a pack of practice problems. So I think those of you that aren't here, don't have to do ISTEP testing, it might be wise to take some time to start working on these practice problems. So I really suggest you try to have this packet finished before we take the test. So I thought I'd hand those out to you now. So I'm not sure, and this is kind of throwing a wrench in things. We have at least another week. I think on the Canvas calendar, I have it kind of like possibly skipped in, but it's not going to, probably not going to be next week yet. But it is going to be over solid vision and all this. Yeah, still a lot of practice problems. So I would say after today, we will probably be able to do through 14. So if you want to make a line like after 14 before 15. So 14 is problems, kind of like our doing now today. That's probably, if you have time, and I suggest you use it tomorrow to work through 14 in the packet. So you should have enough knowledge to do those first 14 problems. Because it's all Mendelian genetic stuff up to that point. So put that in the safe place. So, Mendel, almost all science is a little bit being in the right place at the right time, a little bit of luck. So Mendel was able to discover a lot of what he discovered because he was working with a pretty simple system. So for example, peas are pretty genetically simple compared to us. They have less chromosomes, they have less genes, they just have a smaller genotype in general. And all of the traits that he studied were controlled by only a single gene, like by a single protein, either getting made or not being made properly. Okay? And also was lucky that each one of the traits he looked at, each of those genes either had one of either allele, right? They either made a working protein and caused the dominant trait, or they didn't make a working protein, which would cause the recessive trait. So in all those cases that he looked at, one trait was completely dominant to the other. Okay. This is rarely that simple in humans. There are some diseases that are one caused by one gene. But one thing relates to everything else. So the dominant recessive phenotype is very hard to find in a lot of cases. So he was really just lucky that he's picked the right organism and that he picked the right traits so that he was able to see those clear Mendelian patterns. However, some of his rules are still definitely going to apply. For example, the rule of segregation states that alleles are going to separate from each other, right? So you get one allele from each parent. And the law of independent assortment, that genes on different chromosomes or far apart on the same chromosome are going to get mixed up equally. Those are still going to be really important to keep in mind as we go through this stuff. So, first exception to Mendelian genetics is something called incomplete dominance. And so in the case of incomplete dominance, if you're a heterozygote, you have one of each allele. You don't show the dominant trait, you don't show the recessive trait, you show something in between. So in this case, we're looking at carnation color. We're looking at an R gene. If you have both red alleles, you end up with flowers that are red. If you have both white alleles, you end up with flowers that are white. If you're heterozygous, you end up being pink. Okay, so in between red and white. And so if we think about what's happening here with our proteins, okay, again, what do you think? What type of pigment do you think the red allele makes? Red. The white allele doesn't make any pigment. When you have one of each, you make some red pigment, but not as much as if you had both red alleles. And so the result, you end up being pink. Okay? So all of these, again, we're still following the same rules of what alleles get passed to what offspring. Their percentages are still going to be the same, but our phenotypes are going to be different because of the relationship between our genes and the proteins that they make. Okay? So that relationship between genes and proteins helps to explain why we end up with these different phenotypes, okay, instead of just being just dominant or recessive. So instead of using uppercase and lowercase, there's a couple of different things we can do. So we can just use different dominant letters. I don't actually like that. It's not really the way that most upper genetic classes work. So normally what we'll actually do is use superscripts. So for example, you might see R1, R1, and R2, R2, and then R1, R2. Okay, if you're taking an upper genetic class. So instead of using different letters or uppercase and lowercase, we'll use these superscripts. So as we move on to other problems, you'll probably see more notations like that. Since we're not dominant and recessive, we don't use capital and lowercase letters. We use some kind of superscript. Okay. Change this slides to like that. So for now, we're just going to keep using those letters, though. I forgot to change it. So with your group, go ahead and work through these couple of problems. So if you were Mendel and you were doing this cross between red flowers and white flowers, true breeding red, true breeding white, what would your F1 look like in Y? And then what would your F2 look like in Y? Let's go ahead and talk about that. might help to make some Punnett squares out if you're like completely lost. So here's what would happen. True breeding red flowers, so both red alleles. I'll just use the R and W for now. White flowers. Pure white. Homozygous for the white allele. What would be true about the genotype of all the F1? They'd all be heterozygous and therefore 100%. Hey. If you cross two heterozygous individuals, what's the genotype ratio going to be? 1 to 2 to 1. In this case, that's also going to give us our phenotype ratio because each genotype has a unique phenotype, right? So this one red flower would be homozygous for red. Both of these, the 50% pink flowers, they would all be heterozygous. White flowers would be homozygous for the white allele. And so we're going to still see similar patterns from the Mendelian genetics. It's just that our phenotype ratio is going to be different because we have essentially an extra phenotype, right? Our heterozygous individuals have their own unique phenotype. How do you feel about that? Any questions before we move on? Next exception or kind of category of trait are traits that show co-dominance. So in this case, if you have both alleles, it's going to affect the phenotype equally and separately. So you don't show something in between, you're going to show both traits. So again, we're not doing a blended phenotype. It's not something in between. We're literally going to show both. And one really good example of this we're going to spend a little bit of time on is the ABO blood group. So some of you might know your blood type. Might know if you're A, B, AB, or O. Okay, that's a really good example of codominant traits. I don't know why people are like not taking notes on this here and looking at their phones and stuff. It's really annoying me personally, but okay. So the ABO blood type, it actually has 3 different alleles, and here's where you see some superscripts here, right? We have IA, IB, and a lowercase I. Again, there's a relationship between the alleles that you have and the proteins that you make. So if you have IA or IB, these alleles are going to be co-dominant because the A version makes one protein and the IB version makes one protein. So if you have both of them, you're going to make both proteins. You make A protein and you make B protein. What these code for actually are glycoprotein antigens on our red blood cells. Right, so if you look up here, you see what's this yellow thing here? The cell membrane. We think these big purple blobs are. Proteins. What's attached to these proteins? Carbohydrate chains. Okay. So what do the carbohydrate chains on those proteins do for us? They're labeling, they're like name tags, right, for our cells. So in this case, if you have the IA allele, you make one type of glycoprotein A. If you have the IB allele, you make B. So if you have both of those alleles, you have both of those antigens on your red blood cells. Okay. So you have blood type AB. So again, that's what this is saying. If you're heterozygous for A and B, then you're going to have both of those types of antigens on your red blood cell. Okay. These are not the only antigens on your blood cells, so you might also know the plus or minus, the Rh factor, that's a different type of antigen. Okay. But this is just strictly the ABO typing. The recessive, or the little I, is recessive to both. So this is a damaged allele. It doesn't make any antigen. So if you only have this allele, if you're homozygous for the little I, then you have O blood type because you don't make either one of the proteins. You don't make A and you don't make B. So we're going to kind of take a look at this table here. And we'll talk about the first row, and then I want you to talk about the other rows in your group. So if you are a person with A blood type, you could have two possible genotypes, two possible combinations of those three alleles that are only going to give you A antigens on your blood cell. What would those two genotypes be? Yeah, you're either homozygous for the A allele, or, yeah, you guys remember this a little bit maybe. So two possible genotypes will give you a blood type, and as long as you have that A allele, you're going to have what type of antigens on the surface? So your immune system is going to recognize that your blood cells have that A antigen. If you're given B-type blood, what's your immune system going to recognize? It's not yours, okay? So if you have A-type blood, you have what we call B antibodies. So those antibodies, if they see any B antigens, they're going to attack it. They're going to cause it to coagulate and pack up on it and attack. Okay? So if you have A-type blood that you're going to donate, what other blood types do you think could take it? Okay, yeah. So you can only give to, so sorry, I should say anti-B antibodies. So you can only give to individuals that have type A blood or type AB, actually. You can take A or O. Obviously, you can get A-type blood donated to you. That's what you have. You can also get O-type blood because type O blood doesn't have any antigens on it at all, so your body's not going to attack it. Okay? If it has B antigens, it's gonna get attacked. If it doesn't, you're gonna be okay. So go ahead and talk about those other three blood types really quickly. You can kind of fill in those missing boxes there. Okay. I'll go ahead and fill these in here. So if you have B-type blood, again, you have two possible genotypes. You can either be homozygous or you can be heterozygous with the recessive allele, right? As long as you have that B allele, you're going to make type B antigens. And since you don't have any A antigens, you're going to form anti-A antibodies. So they're going to attack any A antigen that they see because they know it's not, your body knows that's not your blood. And so if we think about donation, you could give blood to, not to A, but to B, or you could give it to AB, but you can only take it from B or O. It's kind of the opposite of if you have A blood type. If you have type 80 blood, there's only one possible genotype. You have to have both the A and the allele. You would have both type, oh no, I didn't work. Both type A and B antigens on your surface. And so you're not going to form antibodies to either A or B because your blood cells have both. So your body's not going to attack any blood cell that comes into your body with A or B antigens. And so this individual would be known as a universal recipient. So that means if you need blood, you can take any blood type, either A, B, or O, A, B, A, B, A, B, or O, because you don't have any antibodies that are going to attack the antigen. There's other antigens to worry about besides A, B, and O, but in this case, right, you can take any A, B, O blood type. If you're O blood type, you only have one possible genotype, but you have to be homozygous, recessive. You don't make either A or B antigens, which means that if those antigens are in your bloodstream, you're going to make both anti-A and anti-B antibodies. Okay, so these individuals are universal donors because they don't have any antigens on the surface. So whether you have these antibodies or not, your body's not going to know to attack it. However, if you have O blood type, you can only get blood from other O types. Yeah, because otherwise, if you got any of this blood into you, anything with an A or B antigen, your antibodies are going to attack it. Right? So just kind of a quick lesson on that. I think it's something most of you, or I don't know, how many of you do know your blood type? Only a couple people. If you donate blood, that's something that they'll tell you your blood type. So go ahead and set up this Punnett square really quickly in your notes. Go ahead and check those genotype-phenotype ratios with your group members when you're done. So your genotype ratio is going to be 1 to 1 to 1 to 1, right? You end up with 4 different genotypes. Your phenotype ratio, though, is going to be 2 to 1 to 1, right? Because both of these genotypes are going to result in type A blood. This individual is going to have AB, right? And this individual is going to have B blood type, right? So 2 A's, 1 AB, 1 B. Okay, so that's the kind of problem you're going to need to do for incomplete dominance. Okay, last one we'll talk about today. So the third one that does not follow those Mendelian phenotype and phenotype ratios called pleiotropy. And honestly, most genes are pleiotropic. Because in a lot of cases, one protein that gets made is going to affect a lot of other protein production, proteins that get made. There's a lot of interconnectedness between one protein and another, one gene and another. So this is when one gene affects more than one phenotypic character. Or another way of putting it, one gene affects more than one trait. So a couple examples of this would be dwarfism, achondroplasia. So you don't just have like shortened fingers or shortened toes, but everything is shortened, right? So more than one trait is affected. Or gigantism, acromegaly, it's kind of the opposite effect. And there's a couple of different forms of dwarfism. So this is just one version of it. Of course, here's maybe one of the most famous individuals with acromegaly, Andre the Giant. If there's any Princess Bride fans out here, you'll recognize him. So, something interesting with achondroplasia, with dwarfism. So let's say we're crossing an individual that's it does have dominant inheritance. So as long as you have one dominant allele, you're going to show achondroplasia. You're going to be a dwarf. So an individual that's homozygous recessive would be a normal individual. And so if you're crossing somebody with achondroplasia, a dwarf, with somebody who's normal versus this other situation, we know here that the genotype ratio would be half and half, or right? So half heterozygous, half homozygous recessive. Okay, just like regular Mendelian genetics, you'd have the same genotype and phenotype ratios, right? Half and half, half dwarf, half normal. So far everything looks normal. This is just the same as Mendelian genetics. There's no difference in genotype or phenotype ratios. However, if you cross two heterozygous parents, if you have both homozygous dominant alleles, that's going to end up being lethal. So you're not even going to produce any offspring that are homozygous dominant. Okay, so the reason that messes up the Mendelian ratios then is you would not see this individual being born. So none of the offspring are going to be born with that genotype. And so all you're going to be dealing with is these three. Okay, so if you think about that ratio, then the ratio is going to be 2 to 1. And so you're going to end up with 67 around, right? There's going to be a 2/3 chance of your offspring being a dwarf and a 1/3 chance of being normal. So that ratio is, in fact, going to be different than on the Mendelian one. So that is the notes for today. Couple things. We have about 10 minutes left of class period. If you have not yet finished the quick check 3, which I gave in class on Friday, that's due tonight. I do have a key up here, so if you have done the problems and when you check your work before you leave, you can do that. So make sure that gets turned in tonight. That's due tonight. I'm also going to pass out the next quick check, just in case, because I know some people are pretty good students in here and are done with it, are probably done with the quick check already. We're not done with this section yet, so you can only do the front page. But if you have the quick check 3 done, you can start on the quick check 4. Or sorry, that's quick check 2. Sorry, this is quick check 3. Sorry, I got my numbers mixed up. No. What do you want to do? Yeah, so at the leaf, make sure you get the quick check from Friday because I have my process done and take pictures of it for him tonight. So I would maybe just get that out now. Yeah, we went over the front of it. The front was the dogs. So we did this. We went over this in class on Friday, but then the rest of it was homework, the back side.